is a review guide for EXP 4204 Human Perception for the first exam. So we started off with the question of what is perception. And the simple answer is trying to understand how our brain, which ultimately makes no direct contact with the physical world, is able to use our sensory information in order to understand what's going on out there in the world. We started by discussing two types of perception, exteroception and interoception. Exteroception refers to bringing information via the senses about the external world outside of our body and trying to figure out what's going on out there. Whereas interoception refers to sensory information coming from our body itself and the brain using that to interpret information about our body. We started off with some examples of interoception. So the first one we mentioned was proprioception, sometimes known as the kinesthetic sense, which is a sense of where our body parts are and what they're doing. And we had the example in class where I moved somebody's hand around into various different locations, and after having done that, she was able to say where her hand was relative to her body. And what we know is that this is based on feedback from our skeletal muscles. Our brain uses the information about our muscles as our limbs are being moved around in order to interpret that as motion of our limbs through space. And the idea is that our body is then able to map where our limbs are in location relative to the rest of our body in order to know where our various body parts are. We also talked about equilibrioception, which is another form of interoception. It's a sense of balance and body motion. So this is how we're able to remain standing up straight, or if we're walking on a high wire, we're able to stabilize ourselves. And it's based on three systems: the vestibular system, which is a movement of fluid in the inner ear, which tells us whether our body is in motion in one direction or another; proprioception, which is again the sense of where our limbs are in space; and the visual system, which is apparently most important. We get feedback as we move around as to whether we're moving in one direction or not, and we're able to use that feedback from our visual system to then provide muscle movement to correct any imbalances. Nociception is the sense of pain. Again, this is a form of interoception, which is based on damages to some with the sense of pain due to tissue damage from chemical, mechanical, or thermal conditions. And it's based on these specialized nerve endings that specifically respond to the kinds of stimulation that can lead to tissue damage. And so again, this is an example of a sense and a perception that has to do with internal body states. And we mentioned that there are some others as well: sense of fullness in the stomach, sense of foreign body in the throat. And so we have a lot of information that we're gathering from inside our body that tells our brain what may be going on internally. However, the course that we're going to be covering, the information that we're going to be covering primarily in this course, is exteroception, which is about the 5 external senses, with which we're all familiar. So vision, audition, taste, smell, and tactile. And so that's based on information from out there impinging on our bodies. It's not about the state of our body itself, but rather the state of the world outside of our bodies, and trying to understand what is causing the external stimulation. And so this is much more difficult in a certain sense than interoception, because it requires, as we will see, quite a bit of guessing and inference about what's going on outside that's causing the stimulation. So we mentioned the idea of sensation versus perception, and what's the difference between those. Sensation, as we said, is based on simple measuring devices. I should say simple, not simple in the sense that we understand everything about them, but they're measuring fairly simple characteristics of the physical world. So they're either measuring things like direct pressure or temperature. In the case of vision, they are measuring things like light energy that's entering our eyes. However, we have to take that information and then combine that with our central nervous system in order to interpret what's going on out there in the world. In order to do this, our brain needs to gather information from our senses, and essentially this central nervous system receives information from the peripheral nervous system. Sensory information then makes its way into the central nervous system via the spinal cord and then to the brain, which then does the actual perception. And so perception takes place in the brain because perception requires a fair amount of computation that isn't in the original sensory stimulus. And so we gave some examples of that. For example, we can have a sensation of pressure and heat on my hand, and that interpretation or perception is that someone else is touching my hand. Or a sensation of colored light that will ultimately be interpreted as an apple. So perception is the interpretation of sensation. And if we recall, we did in class, we had people everybody in the class had to try to guess what object was in their hand that was placed by another person, and we discussed the difference, the breakdown between sensation and perception. Sensation was what you were able to directly measure, and that's things like pressure on your hand, temperature on your hand, maybe the extent to which your muscles are have to be flexed in order to hold an object upright. And those are direct measurements, and what we want to perceive, though, is things like the material that an object is made out of, or the shape of an object, or even the actual name of the object that we're holding. And as we said, perception takes place in the brain because the brain must directly infer or guess what is out there in the world based on the stimulation of the senses. And this idea of perception as guessing is one that we'll keep returning to, and it's the idea that the sensory stimulus is inherently ambiguous and the perceptual has to make a best guess as to what is out there. So what we mean by ambiguous is that it's consistent with different interpretations. So for example, we mentioned size. If we see an object on our retina, the size of the object on our retina is ambiguous. It could be, for instance, if we were to see the Godzilla, it could be a really small Godzilla that's really close to us, which is going to make a certain size object on our retina. Or also it could be a very large Godzilla, say the real Godzilla, but really far away. In this case, the point is that the retinal size is ambiguous. We can't just know based on the physical stimulation, the measurements, actually how large the object is. Another example, a little bit more complicated, we mentioned was lightness. So this is the demonstration of why it won't work. It could be a small Godzilla, or a very up close, or a large Godzilla that's far away. Another example that we gave, as I mentioned, was lightness. And so lightness is a measure of how reflective a certain object surface is. And so when we see an object as black and we see an object as white or light gray or something like that, this is actually our brain's way of guessing how light or dark that object is a way of it's our brain's way of guessing how reflective that object is in general. So when an object looks black, that's our brain saying that this object is really not very reflective. And when an object is white, it's our brain's way of saying this object is very reflective. The question is, how does our brain figure that out? And so the simple answer would be, well, measure how much light is currently bouncing off of the object. So say if it's bouncing off a lot of light, maybe it's very reflective. But as we said, the problem with that is you don't know the illumination conditions. So you can't just measure how much light is bouncing off an object and say how reflective it is. You have to know how much light there is in the room in the first place, or in the environment in the first place. As we said, a dark road in the bright sunlight is actually going to reflect a lot more light than a white piece of paper is indoors. And so your brain has to do something considerably more complicated in order to figure out how whether an object is white or black. And as we said again, so the inherent stimulus, the physical stimulation is ambiguous, and your brain has to overcome that. So how does our brain go about doing this? So throughout the course, we're going to be talking about different approaches. One is bottom-up processing, which is processing based on the incoming stimulus itself, also called database processing. So these are things you do without any pre-existing knowledge about this particular kind of object or the particular situation in which you're in. But this is something you would do regardless of any pre-existing knowledge. You run this operation over the object or over the stimulus. Top-down processing is processing based on receiver's previous knowledge, and we can think of this as cognitive factors, and also called knowledge-based processing. So we gave some examples. So we discussed some illusions. In this case, the example of a lightness illusion where A and B look very different, but as we can see, they're actually the same as one another. And what's going on here is there's really a combination of bottom-up and top-down processing. On the one hand, we're getting bottom-up information that tells us that these are in fact the same, A and B. But we're also getting some top-down information on the basis of the fact that we assume that there's a single light source in this case. And so a single light source, if there's a single light source and B is in shadow, our brain makes the interpretation that B must actually be lighter because the bottom-up information is telling us that these are getting the same illumination. That's the actual measured light that's coming off of them. But because we assume that there's a single light source and that this is actually in shadow in this case, we therefore interpret this same amount of light coming off of here, even though it has less light actually, or we interpret it as having less light that's hitting it. We interpret that as being a lighter surface. An example, a more direct example of top-down processing is the case where we first see an object such as this one, and then when we're posed with an ambiguous figure, so this is something that could look either like a mouse or as like an old man, we'll tend to see this as a mouse because we had previously been exposed to this. If we had seen this first, then we might be more likely to see this differently. And we also discussed sine wave speech. And so sine wave speech are examples where speech signal is broken down into a couple of simple sinusoidal sinusoidal sound files. And when those are heard in isolation, it doesn't sound like speech, but then if one hears the original speech from which it's derived, it will sound a lot more like speech. And so that's another example of interaction between bottom-up and top-down. The top-down is the knowledge of what that stimulus is, and then that changes the way that the sensory information is interpreted. You're taking in bottom-up information, but it's interpreted differently based on the top-down information. There's another example of top-down influence on perception. We discussed the case of the rotating mask illusion. So even though this mask is in fact hollow, the interpretation that your brain ends up with is that it's convex, that it's sticking out, even though that nose is really further away from you in depth. It looks like it's closer in depth. And as we mentioned, this is based on a top-down assumption, which is that faces are convex. We know something about faces. Faces in general have this property. So the bottom-up information is ambiguous in this case again as to what the depth information here. And as we mentioned, many illusions are based on ambiguity of depth. And in this case, your brain ends up with the wrong interpretation. And so our conclusion was that we do not perceive the world out there directly. We have this, access to sensory information, which is measurements of properties of the world, and our brain needs to make inferences and guesses about those that are based on bottom-up processing, so processing of the actual stimulus, but also based on assumptions, so top-down kinds of assumptions that allow us to make draw those conclusions and interpretations. Okay, so we move on to lecture 2 with its methods. And the question, how do we study perception? Do we look at the brain, which is the basis, the physiological basis of perception? Do we look at behavior, which is the behavioral output, the outcome of perception? Or do we look at experience, which is our own ability to internally observe perceptual events? So we identified 4 distinct ways that one could study perception. Look at the phenomenological, which is direct observation of one's own perceptual processes, so basically looking into one's own mind. Behavioral, which is the behavioral output in some sort of task in response to a sensory stimulus. Physiological, which is to look at the brain, look at some measures of the brain. And theoretical, which is to try to look at perception in terms of modeling, in terms of some sort of computational approach where you try to simulate or understand, simulate perception in order to understand it. Okay. So we start with the phenomenological approach, advantages. So basically this is looking into oneself to observe one's own perceptual processes. Some advantages are that it's natural and intuitive. It doesn't require any specialized intervention or equipment. But disadvantages are no external validation. We can be wrong about our own perceptions. As we mentioned, high fidelity experiments where experts, audiophile experts, weren't able to judge higher level equipment as being actually being better than less expensive audio equipment. But they thought, when we're told that it was higher end equipment, they thought it sounded better. Blind sight is an example that we mentioned as well. An individual who, in this case, was able to seemingly navigate past obstacles, but even though they had cortical blindness, and this is a well-known phenomenon. In many cases, people who have cortical blindness are able to demonstrate better than chance of visual performance, even though they do not have conscious awareness of any of their perception. So the point is that a lot of perception can actually take place beneath conscious awareness. Behavioral methods we mentioned were psychophysics, which is to systematically manipulate the stimulus and look at behavioral responses. And it's called psychophysics, the idea being that you can set up the parameters of your experiment and look at the output as the experimental result. And we discussed a couple of different kinds of behavioral methods, a couple of psychophysical methods, both quantitative and qualitative measures. So we'll go to qualitative measures first. Qualitative measures are cases where there's no correct answer. So for instance, judging attractiveness or identifying some sort of ambiguous stimulus. In this case, there's no correct answer as to whether this is a person playing the saxophone or face, but one can observe that one can make you can make a judgment at any given moment as to which one you see. And in this case, you can have consensus, for instance, about which one of these is more attractive, but there isn't actually a correct answer per se to this question, so that we can measure things that make differences in qualitative measures, and of course quantitative measures as well, where there is a correct answer. Detection, magnitude estimation, and visual search are examples of quantitative things that can be measured quantitatively where there is a correct answer. So some particular examples: detection. Absolute detection would be, can you detect the presence of a specific stimulus? So, for instance, here this is a case where the contrast is going down as you go from the top to the bottom, and the question could be, at what level of contrast are you able to detect a certain letter being present here? Or similarly, how loud does a sound need to be in order to detect it in a noisy environment? And so both of these cases there can be a correct answer, and then the question is, how well are people able to detect whether there's the presence of a stimulus or not? Difference detection is similar. In this case, it's not the presence of a stimulus, but the difference in magnitude of a certain stimuli. So, for example, how much different do two lines need to be in order to be able to detect that they're different. And this is something that's called a just noticeable difference, or JND, which we can measure for any given type of stimulus or type of sensory modality. We mentioned a specific finding, which in relation to the just noticeable difference, which is Weber's law, which says that the just noticeable difference gets bigger as the magnitude of the stimuli gets bigger. So if you have to tell the difference between two weights, well, the more each one of those weigh, the bigger the difference that'll have to be between them in order to detect that. And that was the book example we did in class, where if we put cups on top of books, because the books themselves were heavy, you needed more cups in order to be able to tell the difference. At least as we finally got the experiment to work, you need more cups to tell the difference between two books compared with just the cups themselves. But as we mentioned, within a sensory modality, the ratio is constant. So what that means is, in this case, if you go from 100 grams of the base unit, the base amount, and then you need to add an additional 2 grams in order to be able to tell the difference. And so that's a date ratio of 2%. And then if the underlying stimulus is 200 grams, then you need to go to 4 grams, because that's also a ratio of 2%. So you need to increase it by 2% of whatever the underlying magnitude is. And as we mentioned, for other sensory modalities, it could be different. So electric shock, it's only 1% difference. In sound intensity, you need a 4% difference. And so across these different sensory modalities, you have different Weber fractions. And as you said, a smaller number means greater sensitivity. You need a smaller percentage difference in order to detect that there's a difference there. One problem with psychophysics data is that there tends to be noise, and the noise can be in the sense that different people respond differently to the same stimuli. So even if you give the exact same stimuli across trials, somebody may respond differently, and different people, of course, will respond differently to the same stimuli. So even if one person is able to detect a certain sound at a certain level of noise, maybe somebody else won't be able to detect that. So there's quite a bit of variability across individuals, and that's why it's not the same as standard physics, where in many cases you can set up the exact same experiment and get the exact same result. And why is this true? It's because brains are noisy. There's differences across brains, and each person's brain is somewhat different. You can be in a different mental state. You could be tired. You could be thinking about something else. You have more experience, less experience as an experiment goes on, and so on and so forth. So to summarize, psychophysics is very valuable because it allows you to manipulate the stimuli and to then observe the outcome of that in terms of objective behavioral responses. So unlike observing one's own inner perception, this allows you to get objectively valid data. So things like reaction time or percent correct. Disadvantages, unlike phenomenology, is that it's not ecologically valid, which means that you're manipulating things in a way that's not natural. So if you have people doing a highly controlled experimental task, then this might actually affect the way that their brain organizes in response to that task, which may not be representative of typical perception. The next method we discussed was biological, so lesion studies, imaging, and single-cell recording, and we just briefly went over some of these. Lesion studies are, of course, cases where there's brain damage to specific reasons, and so we're going to actually look at several of these over the course of the semester. Certain kinds of brain damage can lead to certain specific kind of perceptual deficits, for instance, agnosias, which are specific recognition deficits. fMRI, which is measuring blood flow responses to particular kinds of tasks, and so this can give us some insight into the location of perceptual processes over a fairly rough temporal time period. So it's not precise in terms of time, but it can give you pretty good, accurate location information in terms of hemodynamic response. EEG or ERP is much more sensitive to precision in terms of timing, and this measures electrical activity at the scalp, and so this can be very useful for saying when a certain kind of physiological response takes place, and this directly measures electrical activity that's due to action potentials as they permeate through the scalp, but of course it has less spatial precision, so it's very precise in terms of timing, but not as precise in terms of where that activity is coming from. So advantages to imaging in EEG and ERP is that you're getting biological data, so it doesn't depend on a behavioral response in a way that may involve people thinking about their task or some other kind of psychological factors. But some disadvantages are responses depend on millions of neurons, so there's a very large chunks of the brain, and it's hard to determine whether you can say something with great specificity in terms of how these things are carrying out their operations based on such large populations. So another technique is single-cell recording, which, as the name implies, is recording the action potentials of individual neurons in response to a specific behavioral task. And the basic setup is you stick an electrode into the axon of the neuron that you're recording from, and then you can present stimuli, and then you look for things like these, which are spike trains, which is the frequency of the response of the neurons in relation or response to a particular stimulus. Advantages are that you're getting very precise information about where something's happening in the brain. Some disadvantages are it's not practical for humans generally, unless they're undergoing specific operations, to do this kind of recording, and perhaps more significantly, there's some doubt as to whether the recording of a single neuron can really tell you much about the operations of the brain, which is this very vastly interconnected network. So thinking about a neuron outside of what the rest of the brain is doing may not give us as much insight. And finally, we discussed theoretical or computational approaches to studying perception. And this is to think about perception as being essentially computing what we call a function, a form of computation, which is taking an input, which is the sensory input, and producing an output, which is either what we can think of as the perception or even the behavioral output. So we talked about a function as being something where there's a mapping between some input and some output. An example we gave was orange juice is the input, and the output in this case, oranges are the input and orange juice is the output, and the algorithm is the thing that brings you from that input to that output. So there are many different possible algorithms that could bring you from an input oranges to the output orange juice. And so the mapping is this abstract idea that you go from one input to some output, and the question is, how do you do it? And that's what we can think out in relation to perception, is the input is the sensory input, and the output is either some sort of behavior or perhaps a 3D world made up of objects and space. And so David Marr discussed the idea of perception as computation, thought about the input as a two-dimensional array of light intensities. So basically what the retina is measuring is a bunch of points of light and how much light's coming in. And the output in this case is a three-dimensional world of volumetric objects in space. And then the question is, what's the algorithm? And so the past 50 years, scientists in both human vision and computer vision have been trying to figure out how you go from this sensory input to this output of a three-dimensional world. Some advantages of perception as computation. Well, on one hand, it's very well defined. You know exactly what the problem is. You have to get from here, point A, which is the input, to point B, which is the output. And the question is, how do you get there? And once you're able to solve that, perhaps that will give us some insight as to how it works. Disadvantages, however, are that it requires some assumptions about the output. So David Marr thought that we should model the problem as one of creating a three-dimensional world filled with objects, but somebody else might argue that it really need to do is model the input from sensory input all the way to behavior. So you have to decide what your output is. And even if you can figure out one way to do it, to produce that output from that input, there's the question of whether that's how the human brain does it as well. May not be the correct algorithm. Maybe this is a solution that only a computer would carry out, but one it doesn't reflect the way the brain actually works. I'll start the discussion of topic 3, the physiology of perception. We start by just viewing the fact that the nervous system consists of the peripheral nervous system and the central nervous system. So sensory information enters through the peripheral nervous system and then makes its way to the central nervous system, either via the spine, which is part of the central nervous system, which then goes to the brain, or directly to the brain in the case of, say, audition, olfaction, and vision. The idea is that we have these nerves which carry that information, that the body has to be wired so that information can make its way from even the far limbs, such as your finger, and make its way to your brain so that they can then be processed. We discussed 3 types of neurons: the sensory or receptor neurons. So these are all of our senses involve sensory receptor neurons, and this is afferent information, means information incoming from the external environment, information coming into our bodies, and that information is then sent to the central nervous system. And then we have motor neurons, which are the doers in the nervous system. These are neurons that stimulate muscles to produce behavior. And then everything in between those loops, so the sensory neuron and then and the loop that leads from sensory to motor response, is association neurons. And this is the vast majority of neurons, such as the spinal cord and the entire brain. And the question is, what do you need all these association neurons to do? So we first start off by mentioning that sensory receptors do this they are specialized to be stimulated by external stimulation, physical stimulation, unlike most neurons, which are stimulated by other neurons via neurotransmitters, which change their firing rate. Sensory receptors' firing rate is activated by the physical stimulation, so this could be light impinging on a retina or pressure impinging on a neuron specialized to respond to that in the skin. So this would be a tactile neuron. And so they produce this thing called transduction, which is a conversion of a signal, in this case a physical stimulation, to a different form, in this case a neural response. And this is what the 5 senses do. And this is just a repeat slide. Basically the idea is that you can measure the firing rate of neurons in response to specific stimulation. And that's what you can do is you see the spike train, which is the frequency with which neurons are firing. So here's a sensory receptor that's responding in response to light, and the more light that comes in this case, the higher the frequency of response. And so that's all neurons can do. It's an all-around response, but they can respond more actively in relation to a sensory stimulus. All senses besides the olfactory sense then send that information to the cortex, but they send it through a way station, which is the thalamus. And so the peripheral nervous system sends information either through the spine or through more direct channels to the thalamus, which then transmits that information to the cortex. In the case of olfaction, it goes directly to the cortex. And this raises the question, what are all these association neurons doing? That's where all the information ends up. It all ends up eventually in this mass of neurons we call the cortex, and the question is why. So we went over a couple of simple principles of neural computation. These are principles that allow neurons to communicate with one another and change their firing responses. We talked about convergence, which is when many neurons send a signal to one neuron, and interaction of excitation and inhibition. And these 2 principles allow neurons to do some pretty sophisticated things. And we gave one example here, which is a neural circuit in which these receptors here are getting light stimulus. And what we're doing is measuring the response of this sensory neuron B over here. And as we showed in this case, the light is stimulating sensory receptor number 4, you're going to get a certain response over here. And then as this extends out here, because these are wired up to B in an excitatory fashion, you're going to get a greater response from B. If we extend it even further out, it's not going to change its response because these guys are not wired up to B. And so the brain needs to have those nerves that connect these neurons from one to another. There's no other way for them to find out what the other ones are doing. This is the property of a receptive field, which we will discuss later on. We would say here that the receptive field of B, in this case, is 3 through 5. Sensory receptor number 2 is not in B's receptive field, so it doesn't find out about what's going on there. If we then connect these sensory neurons A and C to B, however, now we have a completely connected network. And now, if we extend this out, this light stimulus further out here, it's going to continue to excite this thing, the response of B even more. However, if we make these inhibitory as opposed to excitatory connections between A, C, and B, then what we will find is this response, as this gets further out, will reduce. And so what we can say here is that there's an optimal response for an optimal stimulus for neuron B. In this case, it's something that's a bar that's in this location over these 3 receptors. And that produces maximal response, which leads to the idea of a feature detector, which are neurons that respond to specific features in the environment. We'll see several examples of this as we go along. So there are neurons that have been found to respond to specific orientations. Specific orientations of bars, even moving bars, or specific lengths of bars. And we'll see Hubel and Wiesel ended up earning a Nobel Prize for discovering certain properties of neurons that are able to do this specific feature detection. And more generally, the idea is that the brain, through all these interconnected neurons, through excitation, inhibition, and convergence, can ultimately be a basic question asker about what the physical stimulation is out there in the world, and use that information in order to make judgments about what it's seeing. So we give this example here of a motorcycle or a bicycle detector, and it's getting information about these various features, and they can either be excitatory or inhibitory. In this case, wheels would be excitatory for both motorcycles and bicycles, and so if you're trying to discern what that object is out there, if you see wheels, that might make you think, well, it could be a motorcycle or a bicycle. If you then see an engine, however, that's excitatory for motorcycle and inhibitory for bicycle. If you see a basket, that's excitatory for bicycle but inhibitory for motorcycle. And the idea is that by asking the right questions in a certain sense, the brain can try to determine what the sensory stimulus is out there. The next thing we talked about was what is it that differentiates the sensory modalities. The senses, of course, feel very different. They seem to provide very different information, but they all, on one hand, depend on the same thing, which is patterns of action potentials. We know that sensory neurons basically just produce action potentials which are the same across the different sensory modalities. So how is it that vision and audition, or vision and smell, feel so different from one another? So of course, the beginning of the answer is that the actual sensory neurons, the receptors are different. So there's different receptors and they respond to different kind of stimulation. So the visual sensory receptors will respond to light. Tactile receptors may respond to temperature. So they're responding to different kind of stimulation. But as we just noted, the activation they produce is ultimately just a neural signal, and that ends up finding its way to the central nervous system and to the brain. So it doesn't answer the question of how these different modalities end up giving us such different kinds of information. Johann Müller proposed that what gave the different senses their different kinds of qualities is the conducting nerve. It's not what's stimulating it, but it's the conducting nerve. So you can tap on your eyes in the dark, and that will produce a light sensation. And his argument was that's because it's the optic nerve that's conducting the information to the brain, and that nerve relays visual information, and so that's has a different quality than, say, auditory information. However, Roger Sperry, in some well-known experiments, showed that it's not what the brain does with information doesn't really depend on where that information is coming from. And he found that if you took a frog and you took the information that's coming from one eye, and that usually ends up in one part of the brain, So let's say from the left eye ends up on the right side of the brain. Then if you take that nerve, cut it, and then rewire it so that the information from the left side is now going to the left side of the brain, what he found was that the frog's behavior was reversed, so that if you presented a stimulus to its left side, the frog would turn to the right and try to chase after, for instance, a fly that was presented to its left side would now chase it to the right. What that suggested is it's not about the nerve itself that's carrying information, because it's not that the nerve tells you whether it's from the left to the right, but rather where on the brain that information ends up. So if the part of the brain that usually gets stimulated by the left eye is now getting stimulated by the right eye, or vice versa, the brain doesn't know the difference, and the brain is going to act as if it's getting stimulated based on the original wiring. And this leads us to the fact that we know now that different parts of the brain are in fact specialized for different sensory modalities. So we know that the visual cortex is very specialized for visual, the occipital cortex is very specialized for visual information. Temporal portion of the temporal cortex is specialized for auditory information, and these are in fact the portions of the brain where the sensory information projects to. So there are connections between the thalamus and those portions of the brain, and that seems to give them their characteristics, so that the visual cortex does seem to be specifically visual. So this confirms to some extent that the idea that the portion of the brain that the information is coming to, rather than the type of information that's coming there. However, as we noted, the more recent findings from developmental literature and findings about brain plasticity suggest that it's not that those parts of the brain are simply just that way because of the structure at birth, but rather the reason they take on these characteristics is because of the input that they get. So if in early development you rewire auditory stimulation to a visual part of the brain, or visual information to an auditory part of the brain, you will see vision-like properties in that part of the brain that normally would have become auditory. And that's why people who are blind from birth will show reorganized brains that do not have what we would think was a visual cortex, but the part of the brain that's visual will now be doing other kinds of processing, such as auditory processing or somatosensory processing. And so the conclusion here is it's really that the sensory input builds the neural structure, and that determines the nature of the perception. The reason why the modalities are different from one another is not because of the conducting nerve, and it's not because it's that portion of the brain per se. It's because the type of input that leads to a certain activation over time ends up structuring the brain differently. And we will see some more specific examples of this as we go along. For the topic of light, we're now moving away from general principles of perception to a specific sensory modality, namely vision. So we start off with the physical source of stimulation for vision, which is light, of course. And so light is a form of electromagnetic radiation created by the oscillation of electrically charged material. So for most of evolutionary history and in most circumstances, light would have come from the sun. But of course it can come from other sources as well. And light has some advantages and disadvantages relative to other sources of information about the world. It's very fast. You can get information almost instantaneously from very far away, and travels in straight lines. This is very important because it gives us very precise directional information, which becomes really critical when considering what vision is for. Some disadvantages of it can't travel through objects, so you can hide behind something and nobody can see you, visually speaking. And of course requires illumination, so when it's nighttime and there's no light source, you're not able to use vision. Light interacts with matter in several different ways. It can be transmitted, like it is through the air. It can be absorbed or reflected, and this is going to the type of material that's engaging with the light is going to determine what properties of the light, how much of that light, and what wavelengths of that light are going to be absorbed and which are going to be reflected. And it can be refracted, it can be bent, and refraction is going to be important when we're talking about the structures of the eye. So there's two basic properties of light that your visual system can have access to, and that's intensity and wavelength. Intensity refers to how much light is coming from a specific location. So as light gets stronger and there's more light, we see that its intensity is going up. And if the light is bouncing off of a surface, what's going to determine that is going to be a function of how much light there is in the environment and also the reflectance of that object. And as we pointed out, these are confounded. You can't really know just by measuring the amount of light coming off of something how much of that is due to the reflectance of the object and how much of it is due to the overall illumination. And of course, light is also, so natural sunlight is made up of many different wavelengths of light. And as Newton found, natural sunlight contains this rainbow of different frequencies, and these are different wavelengths of light. So the wavelength is light travels in waves, and the wavelength is just how long that wave is as it repeats. So the light repeats in a periodic function. It repeats over and over again in these waves as it travels through space. And the shorter the wavelength, then we call that a higher frequency. So it means in a certain amount of space, how many times does this repeat? And so shorter wavelengths will give you a higher frequency. And if we look at the different colors, they're determined by wavelengths. So the reds, for instance, here we'd say these are longer waves, and therefore we would say they are lower frequency. And as we move from red, orange, yellow all the way to violet, this is higher frequency, and they have shorter wavelengths. Visible light, though it covers a wide range of different wavelengths, isn't nearly the majority of possible wavelengths of electromagnetic radiation. It's only actually a small portion of the electromagnetic of the actual wavelengths that come, for instance, from the sun. And it's an interesting question as to what is special about these wavelengths, that those are the ones that we are able to see. The rest of them are invisible. And as we noted, other species are actually capable of seeing, or at least of detecting and using information at light at other wavelengths. So infrared, which is below red, lower frequency than red, can be detected by certain reptiles, such as certain snakes. And ultraviolet, which is just above violet, so it's a higher frequency than violet, can be detected by certain birds. And so there's a certain choice of our visual system that these are the frequencies that are of interest to us and that we are sensitive to. So reflectance is a property of objects as they respond to light, and what happens as light bounces into objects out there in the world is that they absorb some of the light, and then some of the light they reflect back into the environment. And this reflectance property is going to determine the color and lightness of the objects. So color is going to depend on the wavelength of the light that's reflected out. And so if an object is green, that means it's absorbing other wavelengths of light, but it's reflecting the green light. If an object's white, it's pretty much not absorbing any of the wavelengths, and it's sending back all the wavelengths of light. The lightness is how much light is being reflected, and so, as we said, a dark object is going to reflect less light. It means it's absorbing more of the light, and a lighter object is going to be reflecting more of the light. And so, what the visual system wants to know, and this is something we discussed about in relation to the lightness illusions that we saw earlier, is what the visual system wants to know is the nature of the material. It wants to know, is this a reflective material or a non-reflective material? Is this a material that absorbs these wavelengths or those wavelengths? And those are things that are going to remain fairly constant across different conditions, different lighting conditions. And so it tells you something about the stuff that the object is made of, not the specific environmental stimulation right now. Another property of reflectance is matte versus specular reflectance. So matte surfaces tend to scatter light in many different directions, whereas specular surfaces reflect light in more in a specific direction relative to how the light was coming in. So a perfectly reflecting specular surface is going to send light back out in exactly 90 degrees orthogonal to the direction in which that light came. And that means it preserves direction. And so you're going to get lots of differentiation that's going to show you where the light is coming from different directions. Whereas in a matte surface, you can't see a reflection, for instance, in a matte surface, because it's scattering the light all over the place. So the specular surface preserves the directionality, whereas the matte surface does not. Another property that light can tell you is the location, and that's related to what we just talked about in regards to reflectance. Because light travels in straight lines, it's possible to figure out where that light came from. Unlike sound or smell, which is going to disperse and move out in lots of different directions, light, at least once it's moving in a specific direction, is going to continue to move in that specific direction. And so that can provide detailed information about specific locations and what's happening. So if you look at these flowers, you can get the shape of of those objects, because you're able to tell in very high detail where the light is greater or lesser as you move along these surfaces. So this brings us to the basic idea that what our visual system is trying to do is something called inverse optics. The visual system is getting all this light that's entering your eye, and what your brain is trying to do with that information is figure out where did this light come from, what made it have the properties that it does. And another way to think of this is what distal stimulus, so what physical objects out there in the world cause this proximal stimulus, this light stimulation in the eye to occur. So in a certain portion of my eye, I'm getting some dark, I'm not getting a lot of light, and another portion of it, I'm getting a lot of light. So my brain has to figure that out. Is that because perhaps there's two different surfaces out there that have different reflectances? Or maybe it's because one of them is getting more light shined on it than the other due to, say, a shadow. And so this is what our brain needs to figure out. And as we said, inverse optics, as we've been saying all along, it's difficult because the proximal stimulus, the sensory stimulus, is inherently ambiguous. And we discussed several examples of this before. So again, lateness is a good example. So if we look at this object here, there's a difference in how much light is coming from this portion as opposed to this portion. And that could be because these are in fact different colors from one another, a different paint color as it's called, or perhaps because this is directly in light and this is indirectly in light. And that's actually how our brain interprets this. The point is that this very same color change over here may be interpreted as a paint change, as that these have different reflectances. And in this case, that color change is interpreted as being due to those being at different angles relative to the light. And it can lead to illusions such as this, where this lightness of this and lightness of this are actually the same. They actually have the same amount of light bouncing off of it, but they're perceived as having very different lightness. Same thing applies to color. So this ball here is actually the same color as this one, but the surround suggests that there's different light, different kinds of illuminations that are hitting those, and therefore your brain interprets those as being different colors. And we discussed the case of shape as having ambiguity. So directional ambiguity, as we said, we need to use the direction of light in order to figure out the shape of objects, but we're missing the third dimension, so it's not really possible to directly extract the exact location of where light is coming from. Which is concerned with the eyes. So we started off talking about the location of the eyes, and we simply noted that for those organisms that have two eyes, such as humans, there are different configurations where those eyes can be. So in humans, their two eyes are both in front of the head, whereas in other species, such as rabbits, the two eyes are on the side, different sides of the head. It's an advantage to rabbits in that they have a wider field of view. So humans can only see a little bit over 180 degrees to the side, covering the entire expanse of the visual field, but we can't see well behind, for instance, behind where our head is. Whereas rabbits can see much more, somewhere like 270 degrees around. However, humans have an advantage because this overlap of the two eyes, although in some sense you're getting less visual expanse for each eye, across two eyes. However, that does provide a benefit for depth perception, which is something we will discuss later in the semester. And it's an interesting observation that, generally speaking, predators have two eyes more in the front of their head, whereas many prey animals will have them on the sides of their head. And that seems to suggest that having a wider field of view may be more useful if you're being preyed upon, whereas having them in the front of your head in depth perception may be more useful if you're a predator. We talked about the basic eye structure. and we talked about how light gets into the eye. So it comes in through the pupil, which is a hole in the iris. And the iris is a muscle that can contract or expand in order to let more or less light into the back of the eye, which is the retina. So light travels through the pupil into the back of the eye, retina, which is the layer of photoreceptors that actually does the transduction, which we will discuss soon. The entire eyeball is called the sclera. Let me see if we have some more here. So the sclera is the hard white surface, that the rubbery white surface that makes up the eye, and right where the pupil is, there's a portion of the sclera, which is called the cornea, that is transparent, and that's the thing that actually is exposed to the external environment. And as we will see, the cornea has the effect of bending light as it makes its way into the eye. And inside the eye, there's past the cornea is this lens, and the lens is controlled by ciliary muscles, which control how curved or not it is. The light enters through the pupil and travels through this chamber. The chamber is filled with vitreous humor, and the vitreous humor's function is primarily to keep the eye in its shape, so that it has this rounded shape, which is something we will discuss in a little bit. The pupil's purpose, as we said, is to allow light to come into the eye. It's just a hole, and the iris changes the shape, or rather the size of that hole. And the purpose of this is when there's more light in there in the environment, because you don't want too much light coming into your eye, because we have these very sensitive photoreceptors that can actually be damaged by too much light. On the sides of the eye, we have orbital muscles which control the movements of the eye, and the eyes can move up, down, and also around in this kind of torsional motion. And we discussed 3 different kinds of eye movements. Saccades, which are these jerky kind of motions that you can control even without any particular motion stimulation. Smooth pursuit, which requires an external stimulus, and then vestibulo-ocular reflex, which takes place when you move your head back and forth. So, as I just said, saccades are these fast, jerky motions of the eyes. Even if you have a static stimulus, you move your eyes around it, and it's in order to foveate a stimulus. As we'll see, the purpose of these eye motions is in order to bring the region of interest, the portion of the image that you're interested in, that you want to get more information from, onto the fovea, and the fovea is a dense packing of photoreceptors in a specific portion. Smooth pursuit is a smooth linear motion, so you can move your eyes smoothly across the visual field, not in the jerky fashion that takes place for saccades. And typically it only is in response to external stimulation, so you know some external stimulus in order to be able to move your eyes smoothly like that. And as we said, the purpose of these types of eye movement are to keep the portion of the visual stimulus that's of interest on the fovea, which is the portion of the retina that is able to do precise location discrimination. So if you stare out here, you're not going to be able to see these letters in the center here, because those are falling in your periphery, not in your fovea. Whereas if you stare here, you should be able to make out all of the letters, and even these small letters, which are now close to your fovea. So where you look is going to matter a lot in terms of what you're able to see. The vestibulo-ocular reflex is a reflex whereby your eyes will rotate in their sockets as you move your head back and forth. And this is based on feedback from the vestibular system in your inner ear that tells your brain that the head is moving, and then it moves the eyes accordingly in the opposite direction to keep them stable. Now, in thinking about the eye, we talked about several different eye types and the evolutionary history of the development of the eye. And as we mentioned, the purpose of the eye is to bring light into the back of the eye, into the retina. And this raises the question of why we need this complicated structure of the eye. And as we mentioned, it might seem possible to simply have a flat patch of photoreceptors in order to be able to detect how much light is out there in the world. And as we noted, there are some species that simply have a flat patch of photoreceptors So certain kinds of worms, for instance, and if you go back in evolutionary history, this was the earliest form of visual system would have been something along these lines. However, as we mentioned, the problem with the flat patch photoreceptors is that you'll be getting light from every direction hitting every single point on the retina, and that breaks down the primary advantage of what our visual system uses light for, which is to be able to use it for directionality, to be able to figure out where light is coming from and how much light is coming from each particular location in space. So in order to get more directionality, you can curve that patch of photoreceptors, and that allows you to detect that there's different amounts of light coming from different locations. So if your purpose is to find out directionality, then curving the surface of the eye is beneficial. So in this case, this portion of the eye, if the more light's coming from down here, you're going to get more light up here than you are down here. And if you keep going, you can close up the eye and leave this kind of pinhole. And as we'll see, that has an even greater advantage in terms of directionality. And as we look at even further stages in evolutionary development and complexity, you end up with a chamber like ours, then eventually with a lens, and even a movable and changeable lens, which is the kind of eye that we have. So what is the purpose of all these structures, and why did they develop over time? So the purpose of the eye, and certainly the human eye, in this complex structure, is to bring the light that's coming from outside the world into focus on the retina. To focus light from selected regions onto the retina. So what we mean by focus is a one-to-one correspondence between locations in the environment and the retinal image. So for instance, if we have an object such as this, a focused image is going to have a one-to-one correspondence, so that each portion of the object is only, the light from that object is only falling on one portion of that imaging device and nowhere else. And the problem, the challenge to focus is blur. If light from some location on an object is falling onto multiple portions of a imaging device, then you have what's called blur, and you no longer have that precision in terms of what's happening and what's the reflective properties of each location on an object. So one solution, let's start off with this problem. Problem that light bounces off in many different locations, all hit the same point on the retina. So light's bouncing off in many different directions off of this object, and those are all falling onto a single portion of this imaging device. Well, the problem is then that part of this imaging device, that could be a retina or a piece of film, is going to be getting information about lots of different locations. So there's no longer this mapping, this one-to-one correspondence between the object and the imaging device. So a solution to that is, first of all, to curve the eyes, as we mentioned. So as you curve the eyes, you get better directionality. So light from here, out here, isn't going to hit this portion of the eye. So you no longer have as big a problem of the fact that light from many different locations are all hitting the same location on the retina. An even more extreme solution is a pinhole. So if you just allow light to go through a small portion in space, a small cut through of an otherwise reflective surface, then you end up with this thing called pinhole optics, which means that in this case, light from this portion of the object is only going to fall on this portion of the imaging device, on the retina, for instance. So light from here is going to get blocked. Light from here is not going to be able to make its way through. Only light from here is making its way through, and it falls in this specific location. Light from down here is only going to fall up here. And so this is called a pinhole. And several species actually use pinhole optics in order to form a focused image. And also there's some cameras, old-fashioned cameras generally, that use this kind of pinhole device. And this has the effect of producing what's called perspective projection. When light has to pass through a specific point in order to reach the back and reach an imaging device, it has the effect of inverting the image. So light from on top is going to fall on below, and light from below is going to fall on top of the retina. And it also means that retinal size is going to depend on distance. As the object gets further away, the image that it produces gets smaller. And these both are direct effects of the fact that light has to pass through a small aperture, in our case the pupil, in order to reach the back of our eye. And this means that our brain is then able to use that fact about the projection in order to use that as a depth cue, something we'll talk about later in the semester. So the reason these train tracks look like they're going away from us in depth is because these train tracks are smaller. The space between them is smaller. The image they're producing is smaller than these train tracks here, which are producing a much wider image. And that's our brain interprets that as the can lead to certain illusions of size. So, for example, in this case, the upper monster looks bigger and scarier than the lower monster because our perspective projection is telling us that it's further away in depth because of the converging parallel lines. And the same size image, if it's actually further away, is interpreted by our brain as being a larger object. Problem with pinholes is that they don't allow much light in. It's very limited in terms of the amount of light that's coming through. And so the solution is to try to gather light from the same location on an object and converge that light and bend it back such that you're getting more light coming from that object falling onto the same point on the imaging device. This is really quite similar to what a telescope or a microscope or a magnifying glass is doing. So we have these lenses in our eyes, both in the form of the cornea and the lens itself, which is inside, that can bend the light from the outside into a focal point onto the imaging device. And so the basic idea is to preserve that one-to-one correspondence, but be able to get more light. So lenses are defined in terms of the refraction, how much they refract light. And so a convex lens, for instance, refracts light inward. A concave lens would reflect light outwards. The cornea and the lens are both convex lenses that can refract light and bend it inward. And our lens, in particular, is able to change its shape so that it can be made thicker or thinner using the ciliary muscles in order to change its refractive power. So a weak convex lens is going to bend the light less, and the focal point, the point where those rays are going to end up meeting, is going to be further away than a strong convex lens, which is going to be able to bend that light more. And the reason why this is useful is because some accommodation is useful because changing the shape of your lens is going to be necessary when trying to focus on objects that are further or closer. So an object that's nearby, its light rays are going to need to be bent more in order to bring them to a focal point on the retina. Then an object that's far, it's not going to need to be bent as much. So our ciliary muscles are going to change the shape of the lens depending on how far away or close an object is that we want to focus on. Focus disorders occur when the focal point, the point at which the light is being converged on the retina, is incorrect. In the case of myopia, for instance, the eyeball grows too long in most cases, and the light is being bent actually too much between the cornea and the lens. The light is being bent too much so that it's forming a focal point before it reaches the retina. And what that means is that light will then again split, and by the time it hits the retina, it's falling. The light from one location out there in the world is falling on the two different parts of the retina, and this is going to lead to blurring. In the case of farsightedness, the eyeball is too short relative to how much the light's being bent. It means we can't bend the light enough. So in farsightedness, for instance, when an object is really close, our lens, typically because of aging, isn't able to bend the light enough, which that means is that the light is actually not being brought to a point on the retina. So again, this is going to lead to blurring because the light from the same location out there in the world is going to be falling on two different portions of the retina. So the way to correct this is to have corrective lenses that are outside the eye. So if the light is being bent too much, like in the case of nearsightedness, the solution to that is to have a concave lens which bends the light outward before that light is sent inward and bent inward by the lens and cornea of our eye, and vice versa. For farsightedness, if the eye needs a little help bending the light enough, then you put a convex lens which bends the light inward, and you put that in front of the eye, so that by the time the light hits the retina, it's going to be focused to a single point. Start the final topic of the first exam, which is the retina. So the retina, of course, is the layer of in the back of the eye that actually is involved in the purpose of the eye, which is the transduction of the light stimulus into a neural signal. The retina forms a bowl-like structure in the back of the eye, and encompassing most of the back of the eye, and reaches all the way around to approximately 270 degrees around the eye in the back. Now, the retina consists of 3 layers, and so you have the photoreceptors, which are the specialized neurons that do the transduction of light into a neural signal. Those photoreceptors send the signal to bipolar cells, which then send it to ganglion cells. And the ganglion cells have their axons, which are called the optic nerve, that then actually leave the eye, in the back of the eye, and get sent on to the brain. So you can actually see the layers here. Now, as we mentioned, this design is somewhat backwards in that light has to actually travel through these other layers before it hits the photosensitive photoreceptors, which has some interesting consequences, as we'll see in a moment. So why is the eye designed this way? Partial reason might be that the photoreceptors need to be attached to this layer, which contains this special pigmentation that provides both light absorbing, so that the light doesn't get scattered back onto the photoreceptors, and also provides nutrients to the photoreceptors. So this raises the question of why we don't see the other layers, because they're actually blocking the light from hitting our photoreceptors. So the first simple answer is that they're largely transparent, but they do actually cast shadows on the photoreceptors. So the second reason why is because the shadows that they cast are actually fixed in space. They're fixed on the back of our eye, and they always cast the same shadow on the same portions of the retina. And this leads to something called adaptation. When photoreceptors, or almost any receptors anywhere in the body, get constant stimulation, they start to adapt, and they no longer respond. So if you stare at the red dot here, after a little while, the blue ring will start to disappear because of adaptation of your sensory receptors, your photoreceptors in your fovea. in your periphery. Now, the red dot doesn't disappear because your eye is constantly jiggering around a little bit, just enough that the densely packed photoreceptors, the stimulus that's following them is constantly going to be changing. You can actually see a retinal layer if you do a little trick by holding your hand tight and with just a little aperture and moving it around quickly as you look through it at a white sheet of paper. And this allows you to see the web of the layers, the blood vessels of the other layers in your photoreceptor, in your retina. And it does this because it's quickly changing the shadows that are passing, the light that's passing through those other layers, and that's quickly changing the shadows that they cast on your retina, so it gets rid of adaptation. Another effect of the fact that our eyes are designed somewhat backwards, or the retina is somewhat backwards, is that the neural signal has to leave through the back of our eye. It has to pass through the layers to reach and get it back out, and that means that there are no photoreceptors in a certain portion of our retina, and this leads to something called the blind spot. And you can see the blind spot, and as we saw in our assignment, if you cover one eye and stare at one location in space, then some portion of the image is actually going to fall in the blind spot and will not actually be seen. However, you will fill in that portion of your visual scene. You don't just see nothing there. Typically, your brain fills in that information based on its best guess as to what's actually out there. So the retina engages in this thing called phototransduction, which is the turning of light into a neural signal via those photoreceptors. For every 100 photons that come into the eye, about 50 of them get reflected back out because of the various structures of the eye. So the cornea, the lens, these all reflect light. Even the vitreous humor, the fluid that's in there, is going to reflect some light. By the time it gets to the back of the eye, the actual retina, it's about 50 photons, and only about 7 of those out of 100 get actually absorbed by the visual pigmentation. Now, the visual pigment is made up, is stuff that's this specialized kind of molecular structure that's inside the discs on the photoreceptors. And these molecules contain these long chains of opsin, upon which there are these small little portions that are called retinal, which is the place on this molecule that's light sensitive. And what happens is, this retinal molecule will change its shape through isomerization. So that's the change of shape in response to light. The atomic structure of this thing it gets rearranged so that it changes shape, and this produces a chemical chain reaction throughout the opsin chain. And this keeps building and building to the point where it actually produces a neurotransmitter release. So as little as one photon hitting this retinal portion of the rhodopsin molecule will lead to a chain reaction that ends up leading to neurotransmitter release. There are two kinds of photoreceptors that we discussed. So there are rods and cones. Rods are high in sensitivity, which means they need less light in order to make them fire, but have a reduced spatial resolution. They can't differentiate between different portions of the image that are producing different amounts of light. And they're achromatic. They can differentiate color, as we'll see in a moment. Cones are kind of the inverse. Lower sensitivity, they need more light in order to respond, but they have higher resolution. They can differentiate between different locations. And they're chromatic. They respond differentially to color. So with regard to response to color, the rods and the cones, each one responds differentially to different wavelengths of light. But the difference is that there are three kinds of cones, whereas there's only one kind of rod with regards to how it responds to light. So the cones come in short, medium, and long flavors, and the short cones respond more to blues, the medium cones respond more to greens, and the long cones respond more to yellows and reds. And what that means is that your brain can use the relative response of these different cones in order to figure out what color is out there. So if you're getting more response from the S cones than the M cones, that's a signal to your brain that it may be more in the blue range. And the reverse is true. If you're getting more L than S, that may be something that's in the yellowish range. The rods, on the other hand, only can respond in one way, and so they respond differently. There's only one kind of rod, and so they may respond differently to different wavelengths of light. However, the brain can't decide what the wavelength of the light is, because it's going to be intertwined with how much light you're getting. So let's say you get a certain type of response from a rod. Let's say you're getting a response somewhere in this range. Now that could be because it's a really bright light that's centered in the 500 nanometer wavelength, or it could be a dim light that is actually primarily in the 450 nanometer wavelength. So the problem is there's a confounded the response. There's no way for your brain to know whether a certain response is due to a certain wavelength or a certain magnitude of light. But with the cones, it can use the relative proportion of the different responses of the different cones in order to figure out color. Now the receptors, these cones and rods, are distributed differently across the retina. In the center, in the fovea, this specialized region, that's when one looks at a specific portion of an image, that's the one that falls directly on the fovea, is densely packed with cones. And that's what's seen in the red here, and there are no rods there. And as you get away from the fovea, the number of cones dramatically decreases and the number of rods increases. And to get far out in the periphery, the number of rods starts to drop off, but they maintain rods even way out in the periphery, whereas the number of cones is there's very, very few cones. And this over here, as we pointed out, is the blind spot. And you can see it's a pretty large portion extending over something like 2 millimeters of the retina. There are many more rods than there are cones, 120 million rods, and they're doing all that peripheral vision. But these 5 million cones are what are responsible for central foveated vision. So the rods and cones are different in response in relation to their sensitivity and acuity. So rods are higher sensitivity, as we said, and lower acuity, and cones are the inverse of that. So what is that due to? So rods have greater sensitivity, first of all, because their chemical composition is such that they respond to less light, so they can respond to a single photon, whereas cones need around 100 times that light to typically to produce a response. Also, in terms of size, rods are larger than cones, and so they gather light from a larger region. So that's internal structure, but they are also different in regards to how they're wired up to other layers of the retina. So both the rods and cones are connected directly to these bipolar cells, but the bipolar cells are such. So the difference between rods and cones is that rods are converged on many more bipolar cells than cones do. So this has the effect that rods are pooling light information from a larger portion of the retina. So up to, let's say, 5 rods here are all connected to a single bipolar cell. Well, that means that you can have light, small amounts of light hitting lots of different portions of the retina, and they're going to all be pooled together to produce a response from this bipolar cell. So this bipolar cell is going to have a pretty good sensitivity. The cones, on the other hand, each cone is only going to be wired up to a smaller number of bipolars. So let's pretend each cone here is wired up to a single bipolar. Well, what happens here is that that same amount of light isn't going to make any of these bipolars respond. So this is another way in which cones have reduced sensitivity relative to rods, because they're not sending and pooling their information. So you're not getting as much activation from a smaller amount of light. However, this does lead to the inverse differences in acuity. So if you're pooling light from a bunch of different locations and sending them all to a single bipolar cell, that means you lose the ability to distinguish between what is actually producing the light that's falling on those various photoreceptors. So for instance, in this case, these various rods, these 2 rods are getting activated, or these 2 rods are getting activated. Those are both going to produce the same response from a bipolar cell, because they're all getting pooled together by the time they hit the bipolar. With the cones, though, because these bipolars are separately connected to different cones, you're now able to preserve the location information. So now the brain, when reading off these responses to these bipolars, in this case, with the cones, it's able to say, oh, these 2 bipolars are firing because it's wired up to these 2 cones, whereas these 2 bipolars are firing in this case because it's these 2 cones. And so you get that difference in spatial acuity, whereas here, these 2 responses are indistinguishable, even though they're getting, it's being caused by light hitting different locations on the retina.